Okay, I'm going to get started. I want to welcome everyone. Greetings to all our participants from around the world. I'm Jean Greenberg, President of the International Society for Molecular Plant Microbe Interactions. Today marks the exciting inauguration of our Road to Jeju webinar series. Before we discuss the series in a bit more detail, I want to give a brief update about our society. We recently became self-managed, and therefore we are making a new website. Excitingly, it will be available later this month, hopefully even by next week. The website has many excellent features. It's got an easier to navigate design, cleaner, and there'll be a free job board for members, a community bulletin board for events and announcements. Feel free to send us your events and announcements, and we will post them. You'll also have free access to most of the 2025 Congress presentations from last year. And this will be a hub for all upcoming IS-MPMI activities. You can visit our website or use this QR code. And if you have things to share with us, here's the new contact email.
Yeah, so I'll take over from now. So just to talk a little bit about why we're all here today. So this webinar is part of a journey we've called The Road to Jeju. So as many of you might know, in July 2027, so next year, the next IS-MPMI Congress will take place in Jeju Island, South Korea. So this will take place from the 18th to the 22nd of July. Oh, I think you... there we go.
Sorry.
No worries.
Yeah, go on.
Yeah, so from the 18th to the 22nd of July next year. So for those of you who have been to an IS-MPMI meeting before, this is our flagship meeting, and it brings together more than 1,200 researchers from all over the world to share the latest in molecular plant microbe interactions. So if you haven't already, please mark your calendars for the next meeting. But the thing is, July 2027 is a long way away, so to build momentum on the way there and to keep our global community connected and engaged in the meantime, we decided to come up with this webinar series. So this webinar series will run every 6 to 8 weeks from now through to May next year, and it will alternate between two formats. So the first is Legends of IS-MPMI. So this will feature in-depth talks from leaders who have shaped our field. And then the second format is Rising Stars of IS-MPMI, and this it will be a showcase for outstanding early career researchers who represent the future of our society. So every session is free, open to the public, and will be held live on Zoom, much like this one. But you will also be able to find them later on our YouTube channel and also on our website because they are all recorded. So please help us spread the word about this new webinar series. She can, yeah, there we go. So this is the Legends of IS-MPMI lineup. So we're kicking off today with Roger from Indiana University Bloomington, Roger Innes. And honestly, we could not ask for a better person to launch the series. But looking ahead, we will have Jonathan Jones from the Sainsbury Lab in Norwich, UK in September. In January next year, we'll have Jane Parker from the Max Planck Institute for Plant Breeding Research in Germany. And then we will close out the Legend series in May with Sinyan Dong from Duke University. So the exact dates for Jane and Sinyan are still being confirmed, but we will announce them soon. And you can mark your calendars for Jonathan Jones' seminar on September 30th. So these are 4 genuine leaders of our field throughout this next year, and we hope you will all join us for all of these Legends of IS-MPMI webinars. And then moving on to the Rising Stars format. So alongside the talks from the legends of our field, we also aim to highlight the next generation, right? So the idea of this Rising Stars series is to have a showcase for outstanding early career researchers. So this includes PhD students and postdocs. And this essentially is open to PhD students and postdocs who are IS-MPMI members. These people are invited to self-nominate. I mean, the webinars themselves are open to all, but if you want to self-nominate and potentially give a talk at this webinar series, this is open exclusively to IS-MPMI postdoc and PhD student members. So then from these self-nominations, our board will select the speakers based on scientific merit and based on contributions to our IS-MPMI community. So there will be 3 Rising Stars sessions across the series. The first is in July 29th this year, and then there will be another one in November and a final one in March. So just in case you were not aware, nominations for this first session in July are currently open. So you can self-nominate via the Google form that you can access via the QR code here on the slide. And the self-nomination form will close on June 30th, so at the end of this month. So if you or someone in your lab is interested in potentially giving a talk and sharing your science with the global IS-MPMI community, please self-nominate. So importantly and excitingly, there's real recognition attached to participating in this series. So from all the Rising Star speakers throughout the whole series, the board will select, firstly, a Get Genome Rising Star awardee. So this awardee will give a named plenary talk at the next Jeju Congress, and they will get free registration and travel support courtesy of Get Genome. And secondly, we have the Tannon Excellence Awardee. So there's a second award. And this awardee will receive free registration and travel support to the Jeju Congress, thanks to Shanghai Tannon Life Sciences. So one important point, I did specify that self-nomination is exclusively open to IS-MPMI members, postdoc and student members, but early career researchers based in the Global South may apply regardless of membership status. This is very important. And if you are selected, you will receive a free 3-year IS-MPMI membership. So we particularly want to encourage participation from Global South scientists. So please nominate yourselves and encourage your students, postdocs, people you know in the field to do the same. You can, as I said, scan the code on the screen, or if you can't access a Google form, you can just email contact@ismpmi.org, the email address on the screen, and we will send you a form that you can fill out. Yes.
Today's webinar is made possible by the 4 sponsors shown here. You can use the QR codes and web addresses to learn more about them. Thanks to the Shanghai Tannon Life Sciences Company for sponsoring the Tannon Rising Star Excellence Award. Thanks also to the nonprofit organizations, the British Society for Plant Pathology and the New Phytologist Foundation, which is a not-for-profit organization committed to the excellence and dissemination of knowledge and research in plant science. The foundation currently fulfills this objective by publishing 2 scholarly journals and by funding a wide range of activities such as this one. Finally, thanks to Get Genome for sponsoring the Rising Star Award. Now, I'd like to introduce James Canham, who is representing Get Genome.
Thank you, Jean. Hello, everyone. On behalf of everyone at Get Genome, we're delighted to support the Road to Jeju webinar series. What excites us most about this initiative is how closely it aligns with our own mission to connect researchers with opportunities and to reduce barriers to participation. So alongside this fantastic initiative, we are launching an MPMI themed open call. We're going to be supporting researchers in under-resourced regions to access genomics and accelerate their plant pathogen projects. So if you'd like to know more about that or contribute, please do reach out to us at Get Genome. I want to thank IS-MPMI once again for creating this wonderful opportunity, and we look forward to seeing many of you over the coming months on the road to Jeju. Thank you.
Yes, so I did talk a little bit about Global South researchers in the previous slides. So I want to take a moment to talk about something the board is really excited to share with you guys. So this is our Global South membership initiative. So our colleagues from Africa, Latin America, and Middle East, and lower and middle income countries across Asia are significantly underrepresented in IS-MPMI, and we want to change that. And we're putting concrete steps behind it. So what we're offering is, first, if your primary affiliation is in a low-income country, IS-MPMI membership is now completely free. Second, if you're based in the broader Global South, you can join at 50% of the standard rate. And the third thing, and this is really special, so our president, Jean Greenberg, who you've heard from just now, is personally covering membership for the first 25 PhD students and postdocs from the Global South who request a waiver through the president's Flash membership initiative. So that's a free 2-year membership, first come, first served. Just email contact@ISMPMI. So the bottom line is that wherever you are in the world, there's now a place for you in our society, and please help us get the word out on this initiative. So over to you, Jean, again.
Thank you. I want to just say a word about the Michigan Travel Fund. We are fundraising to help early career members attend the Jeju meeting. And for this, we have the ambitious goal to raise $40,000. We've raised $23,500 so far. So if you have the means, please donate. You can go to our website to do that. And additionally, if you're not a member of IS-MPMI, please consider becoming a member. Your membership ensures that we can cover our basic operating expenses. Now I'd like to introduce Junior Board Member Minu Singla-Rastogi, who will introduce today's speaker.
Finally, our wait is over. Thank you, Jean. I'm very pleased to introduce the former president of IS-MPMI, Professor Roger Ines, who is also the 2025 IS-MPMI Lifetime Achievement Award winner. He is also a member of U.S. National Academy of Science, and Professor Ines has dedicated his career to uncovering the molecular mechanisms of plant microbe interactions. From early work on rhizobium signaling to groundbreaking discoveries on disease-resistant genes in Arabidopsis, his research has shaped the foundational understanding of how plants detect and respond to microbial threats. Roger's work has led to major advancements in the field of NLR's God model of immune signaling, and more recently, the emerging field of extracellular vesicles and RNA in plant defense. Roger's scientific journey is nothing less than highlighting that science thrives on curiosity, challenge, and the importance of rethinking assumptions. And with this, I invite Roger to deliver his talk on the topic leaf surface RNA, how does it get there and what does it do? And please don't forget to post your questions in the quick Q&A chat box so our moderators can use them at the end of the seminar to moderate the session. With this, over to you, Roger.
Thank you very much for that introduction, Minu, and thank you especially to Jean and the rest of the board for putting this seminar series together. It's super exciting, and also I know how much work goes into this kind of stuff, especially now that you all have gone independent, and so just super appreciative. So also I'm maybe not so appreciative of being called a legend. That kind of implies that I'm getting rather old, but I think the lab is still doing really exciting research, and I'm going to present that today. But I also think one reason so many people have signed up to watch the webinar today is perhaps because of this recent event in my lab. And now I need to get my laser pointer going. Okay, so as many of you know, my lab made science last month, but not for reasons that we normally like to make science. So this article appeared about my lab being shut down pending an inspection by the USDA. You may not have heard, however, that we reopened 12 days later after the USDA completed that inspection. So we're back in business, going full speed. It's been open for over 3 weeks already. So things are returning to normal. But one sad thing that happened as a result of the inspection is we had to dispose of all of the houseplants that we had in our lab decorating our windowsills. And the folks in the lab were quite depressed about this. And then a former lab member saw this in the local news that we'd lost our houseplants and took action. And he sent us this nice gift with this nice note. This is from Brian Rudder, who's some of his work I'll feature in today's talk. And he sent us 4 boxes of Legos of beautiful plants with this note that said, enjoy your USDA resistant lab plant. So since we work on disease resistance in our lab, we're really happy we now have USDA resistant plants decorating the lab. And folks in the lab wasted no time putting these together. And this is our beautiful plant on our windowsill now. So things are good again. So leaf surface RNA, I'll explain why we got interested in that in a moment, but at the start, I just want to give you the heads up that this is all the work of 3 fantastic postdocs in my lab. Minu, who you already met, introduced this talk, but then also Lucia Borniego, who is from Argentina, is very happy this morning, and then Mega. And their results I'll kind of present in order during the talk. So how did we get interested in leaf surface RNA? What brought us to that? And our interest really stems almost 20 years back to this 2006 paper from Ralph Huckelhoven's lab in Germany. And they published these beautiful images of powdery mildew fungus infecting epidermal cells on barley. And when the fungus attempts to penetrate the cell wall, the plant can sense that penetration effort and responds with a strong defense response and forms what are called papilla or sometimes cell wall appositions. It's words for the same thing. And when you look at EM, you see these really complex structures accumulating underneath the attempted penetration site. So here the fungus is drilling down through the cell wall, secreting cell wall degrading enzymes. And the plant is sensing both the pressure probably and the molecules released by these enzymes. And in this cell wall apposition or papilla, there's a lot of membrane that accumulates and a lot of callose accumulates, as well as proteins. And it seems to be dumped in there by these things called paramural bodies. And these are assumed to be derived from multivesicular bodies. But when you look at these by EM, they're really odd-looking multivesicular bodies because the vesicles inside of them are very diverse in size. They don't densely stain by EM stains. And so it really raises the question of the origin of these things. And I'm going to get to that later in the talk. But for now, note that there's clearly vesicles in here that seem to be released into this extracellular space. And so that was an interesting paper when it came out. We didn't really do anything with it at that time until about 6 years later. We were doing our own analysis of what happens at a subcellular level during a hypersensitive response in Arabidopsis leaves infected with Pseudomonas syringae. And this is just one image that we had that shows cell wall and then plasma membrane here right next to the cell wall, and then vacuolar membrane. So those of you who are not used to looking at EM may not realize how thin the cytoplasm is on a mesophyll cell. And this is all central vacuole up here. But we saw these structures here that looked an awful lot like that paramural body that I showed you in the previous slide. And we could see what looked like a vesicle here, and it looked also like there's a disruption in the plasma membrane here. And this got us wondering, do these extracellular vesicles get secreted just as part of a defense response? And at the same time, we started reading about extracellular vesicles, and we learned that they were a really hot topic in mammalian cell biology because they'd been implicated in transport of RNA from one cell to another in both mice and in humans. And that got us wondering whether these vesicles might be transporting RNA from a plant cell to a pathogen cell. And so we decided we wanted to pursue that question. It was a novel question. Host-induced gene silencing had recently been published as an effective way to fight off pathogens, but nobody knew how that RNA might get transferred from a plant cell to a pathogen cell. So we started trying to purify these vesicles, and actually, sorry, I got ahead of myself. So the questions we decided we needed to answer was, do plants actually secrete extracellular vesicles as implied by that EM? And if so, what do those vesicles do? And most importantly, do they mediate host-induced gene silencing? So to answer those questions, we needed to come up with a way to purify the vesicles. And this is where Brian, the guy who sent us the Legos, comes in. So Brian took on this project, even though it was a totally new idea in our lab. Nobody else was working on extracellular vesicles. And he figured out how to purify them from apoplastic wash fluid. And what is shown here is a density gradient that Brian did to really purify the vesicles. And so you're literally floating these vesicles from the apoplastic wash fluid in a gradient of iodixmenol. And he found using this marker protein for extracellular vesicles called PEN1, it's a syntaxin protein, was able to find the fractions at which these vesicles floated. And then he was using this transgenic GFP-PEN1 plant to confirm that he was getting vesicles labeled with PEN1. And I should mention that PEN1 was known to accumulate in papillae, which is why we were interested in using that as a marker for extracellular vesicles. And that was work done in Hans Thordal Christensen's lab. So here, this is just these pooled fractions under a confocal microscope, and you can just see thousands and thousands of little green dots. And if you look at that by EM, you see thousands of these little beanbag structures, which is what these vesicles look like by negative stain because they get dried out on the EM grid. And then if you do cryo-EM, if you have a big screen in front of you, you might even be able to see the lipid bilayer of these vesicles. So we were really excited at that time that Brian had succeeded in getting a highly pure prep of these EVs, as we call them. So since we were interested in host-induced gene silencing, we isolated RNA from that vesicle prep and did small RNA-seq, looking for microRNAs and siRNAs. And this is where our first surprise came in. And this is analyses done by Pat Baldrich, who's in Blake Meyer's lab, and then at IU, Ram Potticetti. And what came as a surprise to us is when we looked at the size distribution from our small RNA-seq data. And so from 10 nucleotides up to 34 nucleotides in this graph. And if you look at total RNA from leaves in Arabidopsis, this is the typical pattern. You see lots of 21s and 22s that represent the microRNAs and siRNAs, and a lot of 24 nucleotide RNAs that are mostly from the siRNAs. In our EVs, we saw very few in that size range. Mostly, they were much shorter, like 17 to 10 nucleotides. So we called these tiny RNAs. And the sources of these seemed to come from all over, but by far the major source was ribosomal RNA. And so it looked honestly just like degraded RNA. Interestingly, the supernatant had much more of the 21s and 22s and 24s, and then had a high abundance of this 32. And we know now that this is mostly tRNA halves and the 3' end of 5S RNAs. So this was kind of our first hint that maybe EVs are not the primary source of siRNAs in plant microbe interactions. So this then inspired us, and especially Hannah, who's a grad student in the lab at the time, to just look at RNA in the supernatant versus the pellet that contained the EVs. And this is now a 15% polyacrylamide gel just stained with cyber gold. So we're just looking at our RNA prep. And when you run such a high percent gel, this is what total leaf RNA looks like. So most of your messenger RNAs are going to be up in the very top of the gel here. Total RNA in the apoplastic wash fluid looks like this. So lots of bands here that probably represent just degradation fragments of ribosomal RNAs. Big fat band here, just 33 nucleotides that corresponds to those tRNA halves. When you spin 100,000 times G to pellet all the extracellular vesicles, you find the supernatant is where most of the small RNAs are. So that's consistent with our small RNA-seq data. Interestingly, there's a lot of these long RNAs, ribosomal RNA fragments in the P100. And so this really raised questions about how does all of this long RNA get out there? Why is it even stable in the apoplast? We know there's RNases in the apoplast. And does this RNA have any kind of function? And that's really what we've been spending almost the last 8 years now, really trying to figure out. So to really kind of push at how this RNA is getting out, our first thought was we should do a genetic screen, because my lab really comes from a genetics background in Arabidopsis, to see if we could find Arabidopsis mutants that fail to secrete RNA. And our idea for doing this was to generate a transgenic line that expressed a hairpin RNA that would target a critical gene in a fungus. And the fungal pathogen we like to work with is Calototricum higginsianum, which causes anthracnose disease. And we targeted this SGE1 gene because it was known to be one required for Calototricum to infect Arabidopsis. And also we knew that it regulated a number of toxin biosynthetic genes. And this was work from Richard O'Connell. And so Hannah generated this hairpin construct in which she took 3 separate 100 base pair fragments from different parts of this SGE1 gene and made a hairpin construct that should fold up into a hairpin, which then should induce dicers in the Arabidopsis cells to chop it up and produce these siRNAs. And the idea for the genetic screen is these plants should be resistant to Calotoctricum, and if we could find an Arabidopsis mutant that was now susceptible, that might be defective in RNA secretion. Well, generated the plants, they were resistant to Calotoctricum, but once we got to an M2 generation for doing mutant screening, we discovered we were isolating a lot of plants that had silenced the hairpin rather than bona fide mutants. So sadly, that forward genetic screen didn't work, but this construct turned out to be quite informative for us, and I'll talk about that in the next few slides. So why SGE1? So I said this was really based on advice from Richard O'Connell, who's been a longtime collaborator of ours, and he really developed the Calotoctricum higginsianum Arabidopsis system. And so Calotoctricum is an interesting fungus because it's hemibiotropic, meaning it spends the first 48 hours in a biotrophic phase. So it germinates on the epidermal surface, so on pavement cells, forms an opressorium, and then starts drilling down through the cell wall. And by 40 hours, it forms these biotrophic hyphae, which are fat, and they really displace a large portion of the volume of the cell. And then after about 60 hours, it starts forming necrotrophic hyphae and starts secreting toxins that will kill adjacent cells. So that's the necrotrophic phase. When you delete SGE1 in this fungus, it no longer causes disease. This is what the Arabidopsis plant looks like 6 days after being sprayed with Calotoctricum spores. And under the microscope here, 3 days post-infection, you can see these dark opressoria and then lots of biotrophic hyphae, these fat hyphae inside the epidermal cells. Delete SGE1, it can still form opressoria, but it can't form biotrophic hyphae. So it's required to express the enzymes that allow it to drill down through the cell wall. So this is why it was a target. So the key result that really got us interested in leaf surface RNA was this result from Brian. And so what he did is he took Hannah's transgenic Arabidopsis plant expressing this SGE1 hairpin and just inoculated it with Calotoctricum and as a control, a non-transgenic wild-type Columbia plant. And this is at 48 hours after inoculation, and you see quite a few of these biotrophic hyphae. They're stained here with trypan blue. And you just don't see any biotrophic hyphae on the hairpin plant. And, this is the same Calotoctricum strain on both. This is cotyledons here, I should say. And so this was striking because one, it showed Higgs works in this system, but two, it works really early. We don't have any biotrophic hyphae, yet the RNA must be getting into these fungi. In fact, these fungi appear not to have penetrated the cell wall at all. So this suggested that maybe the hairpin RNA or siRNAs were getting onto the leaf surface, which was not something we'd have considered before we saw this result. And so this inspired us to just look at the leaf surface to see whether that was indeed the case. Got ahead of myself by one slide. To confirm that this was probably true, that this RNA was getting on the leaf surface and then was being taken up to silence the SGE1 gene, Minu did this experiment, which is also quite exciting. She used a strain provided by Richard O'Connell that expresses a reporter gene for SGE1. So this is a SGE1 dependent promoter that expresses mCherry. So when SGE1 is active, mCherry is expressed. When SGE1 is inactive, you do not see mCherry. And so this reporter accumulates in opressoria. And so what is shown here at, again, about 48 hours after inoculation is images of opressoria, these dark spots. So there's 11 opressoria in this image, and 9 of the opressoria are glowing bright red here. So that's just showing that in our wild type control plant, we're getting good expression of SGE1. Now on the hairpin line, again, we've got 9 Oprossoria in this image. Very few of them have bright fluorescence. Several have almost no visible fluorescence at all. And the ones that do fluoresce are quite a bit weaker. And Minu's quantified this, and there's a very significant reduction in that reporter. So this tells us that SG1 itself is being silenced on these hairpin lines. So that inspired then Minu to develop a way to isolate RNA from the leaf surface. And this was a collaborative project between Minu and Lucia. And it's pretty simple-minded. So simply spray the Arabidopsis leaves with a little bit of buffer. We use the same buffer as what we use to isolate extracellular vesicles, so we call it vesicle isolation buffer, and just a tiny bit of wetting agent to help the buffer spread out over the leaf surface. So spray the rosettes, cut the rosettes off, put them into an empty syringe, and then put the syringe inside a 250 ml bottle and spin very slowly. So it's a very gentle spin. And from that, we collect what we call the leaf surface wash. So this is a bit like washing your lettuce and then spinning it in a salad spinner. Take that leaf surface wash, filter it, and then take that filtered water and isolate RNA from it. From this exact same rosettes, then, you can then move on to a vacuum infiltration, apply vacuum, infiltrate the apoplastic spaces, and then recover apoplastic wash fluid by spinning at a higher speed, 600 times G. And again, filter it, and then precipitate the RNA and look at it. And then you can compare apoplastic RNA from leaf surface RNA from the exact same leaves. So this is what they did. And this is what the RNA looks like on a 15% polyacrylamide gel. So again, here for comparison, cell lysate RNA from the same plants, apoplastic wash fluid RNA, leaf surface wash RNA, 3 biological replicates. And what you see is there's lots of RNA on the leaf surface. And this is RNA isolated from the same amount of plant material, from the exact same plant material. And you can see we get just about the same amount of RNA from the leaf surface as we do from the apoplastic wash. And notably, the leaf surface RNA is much less degraded than the apoplastic wash RNA. And so we think this is because there's just fewer RNases on the leaf surface than in the apoplast, because plants do actively secrete RNases into the apoplast. But this finding also indicates that that RNA is probably getting there directly from the epidermal cells, as opposed to like being squirted out through the stomata from the apoplast. Because if it was coming from the apoplast, it should be just as degraded, and it's not. Now, we're curious what was happening here in the higher molecular weight range. So we ran a 10% gel, and you can see here that there are many more larger molecular weight fragments here in the leaf surface wash, just again showing it's less degraded. And notably, if you look at this band at 80 nucleotides, this corresponds to full-length tRNAs. So there's lots of full-length tRNA on the leaf surface. Those are almost all chopped up into tRNA halves in the apoplast. So again, just indicative of more RNases in the apoplast. Okay, so this then brings up the question, why do plants secrete RNA onto their leaf surface? And we, of course, hypothesize it has something to do with defense or simply establishing a healthy microbiome. And so to kind of test that hypothesis indirectly, Minu sprayed plants with salicylic acid to induce defense responses and just asked the question, does that affect the amount of RNA in either the apoplast or the leaf surface? And she saw about a 3-fold increase of RNA in the apoplast that could be recovered from apoplastic wash fluid just 1 hour after spraying. So it's quite a rapid increase in RNA in the apoplast, and a similar increase on the leaf surface here, about 2-fold, we estimate. We also tried desmonic acid and saw the same impact. Again, 2 to 3-fold increase in RNA within an hour of hormone treatment. So this seems to be an active defense response in the plant. So we started all this because we're interested, of course, in host-induced gene silencing. So we wanted to know whether we could detect hairpin RNAs or siRNAs on the leaf surface. And so to answer that question, Minu actually did a transient expression of this same hairpin RNA in Nicotiana benthamiana. And this just shows the total RNA from a on the cyber gold staining, and then this is now a northern blot using a DIG labeled probe. And interestingly, she could see the expected siRNAs in total cell lysate, but we did not see those same bands in the leaf surface wash. Instead, we saw a 300 nucleotide fragment, which corresponds to the stem of the hairpin. So we interpret that to mean that the hairpin RNA is getting secreted on the leaf surface, but the single-stranded region is getting removed, degraded, and it's leaving just the double-stranded region. And in fact, we could see that same 300 nucleotide band with both strands used as probes. So it's sense strand and anti-sense strand probes. So our current interpretation is that when you express a hairpin RNA, the whole hairpin gets secreted, and then what's stable on the leaf surface is the double-stranded bit. Okay, so that really brings us to a fundamental question, which is how does that RNA get to the leaf surface? What is the source of that RNA? And this is totally unpublished data I'm presenting now, and we really think the data is pointing to that the source is really coming from the vacuole. And so this is all work now by Lucia, and this is just the first of many beautiful images I'm going to show you, looking at, in this case, we're looking at a mesophyll cell in an intact leaf or cotyledon that it's expressing a probe called GFP gamma tip. So the green here is the tonoplast, and so gamma tip is an aquaporin that localizes to the tonoplast and is commonly used to label the vacuoles. This here is the cell mask orange, and so up here is your guide, and then the red is chloroplast. So here you can see the tonoplast on one side of the chloroplast and the plasma membrane on the other side. So watch this arrow here. This is actually a movie. Let's see if I can get this to show. There we go. So if you look right here, you can see the tonoplast membrane is forming the structure. And there's actually a name for these. These have been reported previously. They're called vacuolar bulbs. And they're from an invagination of the tonoplast membrane. How these are formed is not understood. But these things can actually break off from the tonoplast and move through the vacuole. And they contain bags of cytoplasm. And so we think these are grabbing RNA from the cytoplasm and protecting it from the RNases in the vacuole. And so if you watch this movie over here, we're going to zoom in. This is just magnification here of what you saw over there. And as we move through, you can see it almost looks like it's pinched off here. And if you keep going, it seems to now merge back. So just going back and forth here. So we think this is actually a way these bulbs get secreted. So here are some still images that kind of confirm that conclusion based on that movie. And so again, we're using cell mask orange to stain the plasma membrane, GFP gamma tip to stain the vacuole, and then red is chloroplast. And so here in this set of images, this is cell mask orange staining. This is the apoplast down here on the bottom of the slide, vacuole on the top of the slide. So these are bulging out into the apoplast. And so it's stained with the cell mask orange, not surprisingly, because it's accessible now to the cell mask orange. But most importantly, you see the GFP gamma tip is getting pitched out into the apoplast. And here, that's just the merge of the two. And this is just another example here. Now we've flipped apoplastas on top. And again, you see that tonoplast marker getting pinched out into the apoplast. So we think this is what's carrying RNA outside of the cell. And here's a super close-up. Again, this is apoplast. And here, this has some extra structure in it. You can see these sort of internal vesicles that would appear in this larger vesicle. And then last, I'm going to show you this movie. Watch this vesicle here. Again, this is apoplast. This is one mesophyll cell. This is another in an intact leaf. And notice this kind of strand here connecting this to either side of the apoplast. So this is really intriguing, how that structure could be formed. And this is a time-lapse video is what you're seeing here. Okay, so again, these are sort of in vivo imaging. Lucia wanted to see if she could purify these things, and we call them extracellular vacuolar vesicles, or EVACs. So these EVACs are big. They're anywhere from 1 to 5 microns in diameter. And what that means is our previous protocol where we've been purifying extracellular vesicles, we were losing those because we were using a filter typically 0.4 microns, sometimes 0.2 microns, and those would have filtered out these EVACs. And so we changed our protocol, and basically what Lucia now does is she takes apoplastic wash fluid, does a relatively low speed spin, because we think the higher speed at 40,000 G was breaking these things. And we know we could go as low as 2,000 times G and still get these EVACs. And then they're not filtered because we don't want to lose these things. So we basically just take a low speed pellet. And again, we're using GFP gamma tip to label them here. And so if you just look at that low speed pellet, you see all of these beautiful green vesicles. And if you zoom in, this is what they look like. They often have a brighter spot at one point, and we suspect this is where it's been pinched off. So we took these purified EVACs and looked at them by cryo-EM, and we see all sorts of interesting structures. Oftentimes, they appear to be double membranes. So hopefully your screen is big enough to see what look like 2 lipid bilayers here. And that would be consistent with one layer being plasma membrane and the other layer being tonoplast membrane. We often see vesicles inside of these EVACs. So here you see a lipid bilayer inside of a larger EVAC. And when you look close, you can often see what looks like a corona on the surface of these EVACs. And that would be consistent with this being plasma membrane that's decorated with a lot of the glycoproteins. So like arabinogalactan proteins are known to have a lot of polysaccharide. So we're guessing these EVACs are coated with these glycoproteins. So what I've shown you so far, we're staining the plasma membrane with just a plasma membrane stain, but we wanted to see whether they also might contain plasma membrane proteins, especially plasma membrane proteins that have been associated with vesicles. And so Lucia generated some plants that were doubly labeled with a RFP PEN1. So this is one of our EV markers that also accumulates at the plasma membrane, GFP gamma tip. And then when you look at the merge, so this is purified EVACs, you can see that this one has what looked to be PEN1 labeled vesicles inside of it. Sometimes they don't appear as vesicles. Sometimes it's just kind of equally distributed, PEN1 and GFP gamma tip. But the important take home here is many of these EVACs are labeled with both an EV marker and the tonoplast marker. And so we think this may be a way that extracellular vesicles get secreted in the first place. And now here, this is with TET8, another well-known EV marker. And we see plenty of EVACs that get doubly labeled for TET8 and GFP gamma tip. And so actually, I'm sorry, here we've labeled the EVACs in this case with cell mask orange deep red. Hopefully this week we'll have labels with GFP gamma tip and GFP TET8. But anyway, purified EVACs from plants expressing GFP TET8. We see GFP TET8 in many of the EVACs. Now, it's not all. Some EVACs have TET8, some don't. Some EVACs have PEN1, some don't. Some EVACs do not have either one. Okay, so in this image, this is, again, a doubly labeled plant where it's intact cotyledon focusing down to the mesophyll cell layer. and doubly labeled with PEN1M cherry and GFP gamma tip. And here the vacuole on this side, apoplast on this side, and we can see one of these evacs getting pinched out. And you can see inside of it, we see that PEN1 label. So this would be consistent with a mechanism for secreting this extracellular vesicle. And here I want to show you a movie of that same evac I'm showing you, and this is a time-lapse movie. So I think you can, let me just go slowly. You can see how that inside that evac, that PEN1 vesicle is moving around. So it's as if it has this vesicle that's floating inside the evac. So again, we hypothesized that these evacs are a mechanism for getting RNA out of the cell. And so, of course, we needed to determine whether evacs contain RNA. And so what Lucia did was to stain it with this dye that specifically fluoresces when it binds to RNA. It's called cytoRNA Select. And beautifully, these evacs seem to be just filled with RNA. And so this, again, purified evacs, just stained with cell mask orange, and with the cytoRNA Select dye, and lots of bright green fluorescence indicating RNA is present in these evacs. Now, as a negative control to make sure that dye was really staining RNA, we collaborated with a lab in chemistry that makes artificial liposomes, and they gave us some of their liposomes that were both empty with no RNA, and then liposomes that had been filled with RNA. And you can see the Cytoselect only stains the one that are filled with RNA. Okay, so we are pretty convinced, at least in our lab, that these evacs contain RNA and that they secrete RNA. But that really raises the question, what does that RNA do? So plants don't secrete RNA so that we can make them do host-induced gene silencing. They must have a reason why they secrete RNA that benefits them in the absence of man-made Higgs. And so to answer these questions, this has really been Mega's project in my lab. And to answer it, we decided we needed to have a slightly more natural environment for addressing it. And so Mega and I decided to do some field work. And I have to show this slide because in my 36 years of investigating plant microbe interactions, I've never done a single field experiment until this one. And so I wanted to prove to people I actually do field work at least once. And so this is a collaboration with Kendall Corbin, who has a lab at University of Kentucky. And she was growing maize out in the field at the University of Kentucky. And we went down there and simply collected maize leaves from, I think these were 6-week-old plants, and isolated leaf surface RNA and apoplastic RNA from these leaves that were growing in the field. And because you might question whether RNA growing under the plants growing under the bright sun, whether there'd be any RNA left on the leaf or not. And so that was one of the reasons we wanted to do this in the field. And so here is, we're just collecting the leaves, keeping them a little bit moist. And then we actually drove all the way back from Lexington, Kentucky to Bloomington, Indiana to collect the RNA. And so Mega, and with the help of Minu and Lucia, isolated the RNA from those field-grown maize leaves and just looked at whether there's RNA on the leaf surface. And in fact, there is. So that's what's shown here, 3 separate plants showing lots of RNA on the leaf surface. It looks a bit more degraded than what the Arabidopsis leaf surface RNA looked like, but nevertheless, there's plenty of RNA there. So Mega took that RNA and then did a standard RNA-seq on it, small RNA-seq on it. And then for the leaf surface RNA in particular, she did a metatranscriptome analysis. And I should note, we did not filter this leaf surface RNA because we wanted to actually capture bacteria on the leaf surface so we could identify what bacteria are on the leaf surface. Because one of our hypotheses that we're trying to test here is that leaf surface RNA plays a direct role in establishing a healthy microbiome. So from that metatranscriptome analysis, Megan identified all sorts of bacteria. It's quite diverse, and quite a few eukaryotes as well. So these are various fungal species mostly. Boxed in red are bacterial species that we decided to use to generate a SynCom, so a synthetic community, to do some direct testing of the impact of this extracellular leaf surface RNA on the growth of these bacteria. And this is just in bigger writing, so you can read it, the genera that we chose for this SynCom. And this was done in collaboration with Kendall's group and a student, Hannah Lefevre, in her lab, that helped actually isolate the bacteria, living bacteria corresponding to this leaf surface RNA, isolated off those same leaves. And so to test whether this RNA might have an impact on growth of bacteria, we took a collection of RNAs. So first, total leaf surface RNA is shown here, and then synthetic RNAs that we identified as being quite abundant on the leaf surface RNA, and just tested whether those RNAs impacted the growth of Pseudomonas syringae strain DC3000. That's just our standard lab strain that we work with in my lab. And very exciting for us, we found that this leaf surface RNA quite significantly reduced the growth rate of Pseudomonas syringae when grown in minimal media. And as a negative control, we just used scrambled RNA that had some predicted structure to it, but had no similarity to tRNAs or ribosomal RNAs. Here, these abbreviations just tell you where these small RNAs came from. These are 20 to 40 nucleotides long. So 16S RNA from mitochondria, 23S RNA from chloroplast, and then these various tRNAs and ribosomal RNAs from the nucleus. These are examples of ones that actually inhibited growth. There were about 32 others that we tested that did not seem to inhibit growth. So this is a sequence-specific effect. So this is, again, on our lab strain. We also tested 4 members of our SynCom. So these were the strains that were isolated from the maize leaves. These 2 are gram-positive bacteria. These 2 are gram-negative bacteria. And here, this just represents all these different small RNA sequences that we had synthesized. So mostly ribosomal RNA fragments, tRNA-derived fragments, a couple of microRNAs, and then some other miscellaneous non-coding RNAs. Every time you see a red asterisk, that showed a significant reduction in growth. And on the x-axis is area under the curve. So that's just looking a standard growth curve, the total area under that growth curve, which is a measurement of growth. So 2 things to note here. Different RNAs inhibit the growth of different bacterial species. So there's a sequence-specific effect that differs from one species to another. Another important take-home here is this Curtobacteria is inhibited by a lot of different small RNAs, whereas microbacteria, at least we didn't see a significant effect from any of the RNAs we tested. So the different bacteria have different sensitivities. And this is then a precursor to test our assumption that the microbiome assembly itself is heavily influenced by the RNA on the leaf surface, because there's different sensitivities by the different bacteria. This result also would indicate that bacteria take up RNA from the leaf surface. And to confirm that, Mega did this nice RNA uptake experiment where she took one of her ribosomal RNA fragments labeled with Cy5 and a tRNA fragment labeled with Cy5, and just incubated it with bacteria in that same minimal media that we use for the growth curve, and asked whether it's taken up. And the way this experiment is done is we incubate the RNA for 25 minutes with the bacteria, and then add a quencher that should block fluorescence of any dye that's still outside the cell. So only dye that's been taken up inside the cell will still fluoresce. And I think you can see many of the bacteria here of Pseudomonas syringae DC3000 are fluorescing bright green, and a lot are not. So what's interesting is it's almost an all or nothing process here with Pseudomonas. Also true for the tRNA. Dye by itself, interestingly, is not taken up. So we don't see any take up of just the free dye. And this is a negative control with no RNA added and no dye. This Cernobacteria is more of a gradient. Some cells are lightly fluorescent, some are brightly fluorescent. And notice this bacteria really tends to form clumps. And so it may be a stage of forming a biofilm that determines how much RNA it takes up. tRNA is a little more evenly distributed. You probably need a bright screen, but there's lots of cells here that are fluorescing, but it's just not as bright as with the Pseudomonas. So the take home here is bacteria definitely take up the RNA, and it seems that some bacteria take up RNA better than others. I'm not showing you the microbacteria result, but we did not see any evidence of uptake by microbacteria, consistent with the lack of effect on growth. So just about the end of my talk. So I want to say this does leave some major questions, what is the cellular source of leaf surface RNA? We hypothesize it's the pavement cells, but there could be other cells contributing, maybe guard cells, maybe trichome cells. And so Minu is currently testing different cell type specific promoters driving a hairpin to see what is the main cellular source of that leaf surface RNA. Are EVACs the primary source of xRNA? We know they contain xRNA, but we don't know if they're the only source. We're trying to identify mutants now that fail to secrete EVACs, and that will allow us to test whether EVACs are really the primary source or not. How does leaf surface RNA get through the cuticle? We really want to figure that one out. We assume there must be channels in the cuticle, and we're now looking at various cuticle mutants to see what effect they have on secretion onto the leaf surface. How rapidly does leaf surface RNA turn over? Our preliminary data indicates it turns over quite fast, because when we wash leaves and isolate RNA, 30 minutes later we can wash leaves again and still get almost the same amount of RNA. And the fact that we saw that huge increase just an hour after SA treatment would indicate it's a pretty fast process. Does Higgs of fungal genes by leaf surface RNA require fungal dicer argonaute proteins? If our hypothesis is correct, that it's those long stems of the hairpin that accumulate on the leaf surface, that would indicate that you'd expect fungal dicers and argonaute proteins to be required. And lastly, we want to show that this leaf surface RNA plays a role in regulating the composition of plant microbiomes. And Mega will be pursuing that experiment very soon, where we take a mixture of our SynCom and look at what effect that has on the relative abundance of each member over time. Okay, so just final acknowledgements. Again, this is primarily the work of Mega, Minu, and Lucia, but of course it wouldn't have ever gotten started without Brian's work on the EVs. And then I didn't present any of Suchi's or Ben's work, but they did a lot of really nice work on the extracellular vesicles. Suchi has some gorgeous EM showing these permeable bodies, which we now think are actually just these EVACs in the process of being secreted. Also need to acknowledge the collaborators and funders. We have 3 fantastic core facilities at IU: light microscopy imaging, EM imaging, and then mass spec. And those are the folks running those centers who have all been extremely helpful. And then this has been long-time collaboration with Blake Meyer's group, Hans Thordal Christensen's group, Richard O'Connell's group, and more recently then with Kendall Corbin's group. And of course, funded mostly from National Science Foundation, but also definitely contributions from the Department of Energy and then Novo Nordisk Foundation in Denmark, who that grant is in collaboration with Hans. And then lastly, of course, thank the folks who are funding this webinar, Get Genome, British Society of Plant Pathology, PEN, and New Phytologist Foundation. And please, you'll probably get an email or a notification to answer a webinar survey, so we would like you to do that. Also remind you to sign up for a Rising Stars presentation if you think you've got some exciting MPMI data to share with the community. And especially if you're not a member yet, please do join MPMI. It helps us put on this webinar series, and I'm happy to take any questions.
Right. Fantastic talk. Thank you, Roger. Please, to the audience, feel free to post or write your questions into the Q&A box, and I will read them out loud. We have already some questions for Roger. So the first one is from Pawan Kumar, and he has or she has two questions. One, what is the RNA profiling of mesophyll cell? Is it similar to the vesicles of the extracellular RNA or different? And the second question is, what is the stage of plants you have used for isolating extracellular RNA? And is there any difference with developmental life cycle of the leaf?
Yeah, good questions. So almost all of our work on the leaf surface RNA has been done with older plants, like 5 to 6 weeks old. And that's mostly for convenience because the leaves, of course, much larger surface area. It's easier to isolate RNA. We've not tried to isolate leaf surface RNA or even apoplastic RNA from seedlings. I did see a paper quite recently on bioRxiv where a protocol was presented for isolation of extracellular vesicles from seedlings. So using that protocol probably could be adapted for isolating RNA, but we've not done that. As far as the profile of the apoplastic RNA and leaf surface RNA compared to the whole cell lysate RNA, what we see is a much higher abundance of what I would call structured RNA, especially in the apoplast, but also to a large extent on the leaf surface. So what I say structured, what I mean is when you map the reads, so like the ribosomal RNAs and the tRNAs, they tend to map to regions that are double-stranded. And that's consistent with that hairpin RNA blot where it looked like it was just the hairpin that was present. I mean, it was just the stem that was present, not the loop. And so our current thinking is that in general, bulk RNA gets packaged into these EVACs, then bulk RNA gets secreted, and then once the EVACs break, and we think they probably get broken pretty quickly, the RNA starts to get degraded, and single-strand RNA gets degraded quickly, and what gets left over is more structured RNAs that are more resistant to RNases. And that's consistent so far with the sequencing data we have.
Right, perfect. Thank you. So we have another question from Roger Weiss. It says, the string-like connections or connectors in the apoplast look like stromules. Is that possible?
Oh, interesting question. That's not one I've ever thought about. So one thing I should explain is the way that imaging is done is the apoplast is flooded with buffer first. And that's why these EVACs are probably floating around in the first place. So if you can imagine an EVAC that's just, in the process of getting secreted and is kind of on the surface of the cell, and now you inject all this buffer, I could imagine there's just, physical forces of that buffer moving around that's pulling on that EVAC. And so it hasn't been pinched off yet. You may see that string. I wouldn't think that it's derived from a stromule, but one thing that is worth noting is Lucia has seen EVACs that have whole chloroplasts inside. It's not common, but you do occasionally see a whole chloroplast inside an EVAC. And that may explain why we do see quite a bit of chloroplastic-derived proteins in EVACs. I didn't describe it, but we've done some proteomics on purified EVACs, and we do see a lot of chloroplast proteins in that proteomics data set.
Wow. Okay, we have another question from Ryan Kessens. And he states, are there conserved sequences or structures shared among the leaf surface RNA types that you're testing? Something that you would explain why random RNA wouldn't suppress bacterial growth?
Yeah. So as I mentioned, that what seems to characterize the RNAs that are most abundant on the leaf surface are ones from double-stranded regions of either ribosomal RNA or tRNA. We think, though, that once a bacteria takes up these RNAs, these double-stranded RNAs may get separated, and there may actually be sequence-specific base pairing with messenger RNAs in the bacteria that then inhibit translation of specific targets. That's just a hypothesis, but MEGA will soon be comparing the proteome of bacteria treated with these RNAs and not treated with the RNAs to see whether there's specific proteins that go down in quantity as a result of that RNA treatment.
Super interesting. We have another question from Kishore Ingol from University of Cambridge, UK. It asks if EVs are increasingly recognized as RNA carriers, but what is the currently known about their proteome? Is there evidence that plant EVs or EVACs also transport defense or immunity-related proteins?
Okay, so let's first distinguish some terminology here, that the way I use it at least. So EVs would just be extracellular vesicles smaller than 200 nanometers, and based on the data I just presented, we think many of these EVs probably get secreted inside these EVACs. So the EVACs would be the bigger vesicle that's like 1 to 5 microns in diameter. So I said we've started to do proteomics on these EVACs, and certainly we do see some defense proteins there. But our general feeling is that probably these EVACs do not survive very long once they get secreted, and it's their contents that are important for defense responses. And what we haven't done yet is to look at whether the contents of the EVACs change after, infection. And we would expect that's probably true, but we don't have any data on that yet. So I guess a long way to answer that question is simply say, almost certainly the EVACs play an important role in defense and probably do deliver defense proteins, but we need to generate that data.
Right. So the ones you have studied until now are the constitutive EVACs.
Correct.
Okay.
Yep.
Great. Okay. Then we have another question from Dennis Halterman asking, some plant viruses can disrupt acidification of the vacuole. Have you looked at virus-infected Arabidopsis to see if there are changes to the RNA on the leaf surface?
So we have done a little bit of that work in collaboration with a lab in Spain. One of their students, Pablo Morales, came to our lab a couple of years ago and was doing infection with Gemini viruses, which is a DNA virus. And he saw an increase in RNA secretion and extracellular vesicle secretion. And they've just posted a paper in bioRxiv if you want to check that out. Their main interest, though, was whether viruses themselves might get packaged inside EVs or viral genomes. And they have some evidence that that indeed happens. But we've not done a follow-up since we've discovered these EVACs to see whether EVACs might contain viruses. I think that's a really interesting area to pursue in the future.
Super cool. Okay, we have another one from Gulshan Thakur. It says, does biotrophic pathogen uptake a small interference RNA or double-strand RNA that targets a pathogen effector through the haustoria or hyphae or by EVs or by any other mechanism or all of them?
Yeah, I mean, certainly we can't rule out to answer all of them, but I based on our work with Calotoctricum, I strongly expect that powdery mildew would also take up RNA from the leaf surface, because it seems like as soon as that spore germinates, it's putting out, this initial germ tube with a powdery mildew fungus, and that germ tube is certainly taking up molecules from the cell surface. So I would expect that gene silencing would initiate right after germination. It probably continues, though, once a haustoria forms.
Super interesting. Okay, we have another one from Babak Benham from UPSC Sweden. A simple and naive question. It says, in the case of tropical climate and high temperature, as RNA is sensitive to heat, how surface RNA will be stable or survive?
But we've not even considered that question. As you know, I keep saying we think the structure of the RNA is probably important to its stability. So I would guess at higher heat, some of these smaller double-stranded fragments might separate. So yeah, I bet you would see a change. But yeah, we've never tried raising the temperature to see how the profile changes. Nina's shaking her head like she's going to do that experiment next week.
She thought about it. Okay, we have another one from Nazari Brandon. Do you think that surface RNA is mostly a biochemical impact on bacteria, like RNA interference, or could there be a physical, a mechanical effect? For example, changing leaf surface charge or impacting addition on leaf surface or other biomolecules?
Certainly it would change the physical characteristics of the leaf surface, as you know, RNA is quite negatively charged. As far as its effect on the bacteria, based on our really simple, in vitro growth experiments, we did not see an effect of our so-called scrambled RNA. So RNA by itself did not seem to affect the growth of the bacteria that we tested. And we thought it might use it as food. We thought it might actually increase growth, but we didn't see that.
Interesting. Okay, we have another one from Barbara Kunkel. Could the leaf surface RNA still in EVACs be secreted from mesophyll cells and make its way to the leaf surface via the stomata?
We certainly can't rule that out. I mean, we know the apoplastic cells are secreting EVACs. Most of those images that Lucia made that I showed you were from mesophyll cells. Yeah, whether they would make it all the way through the stomata, yeah, if they did, I don't know why we wouldn't see a lot of the degraded apoplastic RNA too. I mean, why wouldn't the other RNA that's been released into the apoplast also come out? But yeah, I'm not sure how we would separate those two. It's an interesting question.
Complicated. Okay, we'll read another one. We have many, many questions. Some of them will remain unanswered, but we will send them to Roger and he can email you the answer if you are interested. So there is another from Xing Peng Xie. It says, I have a question regarding the extracellular RNA samples collected from the leaf surface. How do you verify that the recovered nucleic acids are RNA rather than contaminating DNA? For example, did you perform RNase treatment or include other negative controls to confirm that the detected signal is specifically RNA?
Yeah, so we do DNA treatment before we make our RNA-seq libraries. And so at least the RNA-seq data we know is coming from RNA.
Perfect control. Another one from Saskia Hogenhout. Do you think the bacteria and fungi can take up entire EVACs? They may take up the EVACs to acquire nutrition during growth, for example?
That's certainly possible. We haven't tried feeding an EVAC prep directly to the fungus.
Yeah, might happen. Okay, we have another one from Ralph Huckelhoven. Sorry if I pronounced wrong the surnames. The EVACs have to pass not only the cuticle, but first the cell wall of the secreting cell. Mm-hmm. Any hypothesis how that should work, or could it rather come from wounded cells, like in damp signaling?
Yeah, so we've given a lot of thought to that. Our method for isolating both the leaf surface wash and the apoplastic wash is pretty gentle. And then we look for dead cells after those protocols, and we don't see evidence of broken cells. So how do these EVACs get through the cell wall? My guess, and this is just hand waving, is that the cell wall itself is pretty dynamic, and through changes in pH during the response, that you may be getting very localized opening and loosening of cell walls, and these EVACs themselves are quite flexible. They squeeze into hot dogs probably, and so probably can get through relatively narrow channels. So that's, again, it's a guess. It would be really nice to have a way to image that process. But we know they get through the cell wall.
Great. It will be nice to see images of that. So there is another one from Son J from University of Lausanne, an African Genome Center. It says, I have a question regarding the different small RNA sequence impact on different leaf isolated bacteria species. The sequence differences can be considered as a qualitative factor. Can we also know about quantity dependency of the observed regulation for different bacterial species? So the quantity and the quality of the RNA to affect a given bacteria species.
I'm not sure I completely follow
Okay.
the question.
Let's see if I can be clear. The question says, regarding the different small RNA sequence impact on different leaf isolated bacteria species, the sequence differences can be considered as a qualitative factor. Can we also know about quantity dependency of the observed regulation for different bacterial species?
Yeah,
So the amount, I guess.
yeah. So there, if I understand the question right, is the sensitivity of the bacteria to the different RNA species. How dependent is that on RNA concentration is it? And might you see different impacts at different concentrations? Yeah, we've not had time to really dig into that. We selected an RNA concentration based on assaying first the total leaf surface RNA, and we basically were looking kind of for the minimal amount of RNA from total leaf surface RNA required to see an impact in Pseudomonas syringae. And so that gave us kind of a minimal concentration. And then we just stuck with that one concentration for each of these synthetic single RNAs. But certainly I would expect some RNAs are much more potent than others, and that's another good area for future work.
Okay, we have more questions. Hoidin Jo says, could plant-derived RNA be recognized by bacteria as an exogenous RNA signal similar to viral invasion and consequently trigger a trade-off that suppresses bacterial growth? So plant-derived RNA can be recognized by bacteria as an exogenous RNA signal, similar to a virus that I guess a fake that's invading the bacteria, and trigger a trade-off that suppresses bacterial growth.
I'm certainly the bacteria are taking up this RNA. That must mean they sense the RNA somehow. And then I guess the question is whether that itself can affect the growth sort of independently. Yeah, I mean, it'd be hard for us to really sort out exactly why the growth is slowed. Hopefully our proteomics analysis on the bacteria might give us some hints. You know, we see specific housekeeping proteins that get knocked down.
Great. So we have another from Natalia Céspedes from Chile. It says, can leaf surface RNA function as a signaling mechanism to recruit endophytic bacteria into the apoplast?
So I mean, our overarching hypothesis is that this leaf surface RNA is important for establishing a healthy microbiome, and that certainly would apply to the apoplast as well as the leaf surface. And so, I mean, it certainly would make sense to me that if the leaf surface is the initial place where all the bacteria are settling, you know, the ones that can thrive on the leaf surface are more likely to be the ones that get into the apoplast and thrive there. But I think it's well known that the bacteria that thrive inside the apoplast are a relatively small subset of the total bacterial species on the leaf surface. So it may be other factors once you get into the apoplast that determine survival, or maybe specific RNAs are more impactful in the apoplast. There's a lot of work to see.
Fantastic. We have another one. It says, Rao Upapalati says, what challenges you see in taking this knowledge to develop commercial products?
So ultimately, this is the goal, right? So one of the things I did not talk about at all is we do have evidence that this leaf surface RNA is stabilized in part by association with polysaccharides such as pectin and related. And so we're pushing on that side of things to see whether we can figure out what's the optimal way to stabilize leaf surface RNA, and does that complex, we think basically condensates are forming on the leaf surface. And perhaps if we understand how these natural condensates work, that will give us a better idea of how to do things like spray-induced gene silencing. And also, if we know what the RNAs are that really contribute to a healthy microbiome, maybe we can spray that mixture and keep plants healthier that way, essentially through probiotics. So
Right.
wild speculation, but Raul, get a hold of me if you want to invest some money in this.
Cool. So related to your last answer, Chang Sik Ho says, why do you think that plants secretes diverse RNAs to the leaf surface? Why so diverse?
So our current thinking is that there's really not much selectivity on the plant's part as far as what gets secreted. We think it's just grabbing pockets of cytoplasm where there's lots of RNA there, including whole ribosomes. We do have evidence that whole ribosomes are getting packaged into these EVACs. And what we then find on the leaf surface is just the subset of RNAs from that whole cell lysate that are stable on the leaf surface. And then why that would be somewhat selective for some bacterial species over another, I think would be more the result of coevolution. That the ones that do a good job colonizing the surface are the ones that have figured out how to construct their own genomes to work well in the presence of these RNAs.
Right, to avoid that. Okay, we have another one from Sang-Hil Kim, a postdoc in Ping He Lab at the University of Michigan. He says, thank you for your wonderful talk about EVs and its function in plant immunity. And the questions are, what EV cargo is responsible for delaying bacterial or fungal growth? The proteins, the lipids, the metabolites, or associated cell wall materials? And a second question, why are those vesicles not fused, especially when you isolate them at high concentration? They don't fuse between them, I guess it means.
Yeah, and probably don't fuse or based on just charge reasons. I think these EVACs are probably pretty negatively charged. And also you saw in that cryo-EM, they also seem to be coated with a lot of polysaccharide, which themselves are probably negatively charged. And so I think that keeps the vesicles from aggregating and fusing. As far as what has the antimicrobial activity, obviously we're very biased right now, focused on RNA, but certainly they probably also package both secondary metabolites and proteins. But if I had to wager, I would guess it's the secondary metabolites and the RNAs that are the most active in regulating microbial responses.
Okay, fantastic. We have another one. This question is from Corinne Walsh. Do you think the role of leaf surface RNA is largely for defense, or could it also be for curating or promoting beneficial microbes?
Yeah, absolutely both. I mean, we really do think that the ones that colonize the surface and keep the plants healthy is what the plant wants. And so we think these RNAs are trying to promote those bacteria.
Two roles are the same. And we have another one from Ghulam Murtaza from University of Sindh in Pakistan. How do environmental factors affect this, like humidity, temperature, that he asked, or rainwash that washes this RNA pool from the leaf?
Yeah, so we've done this repeated wash experiment and find that RNA seems to just keep coming out. So we think it's going to be relatively resistant to rain. I mean, RNA will wash off, but it gets quickly replaced. Temperature, as I said, we haven't tested that yet. That's really interesting. Abiotic stress is based on the sensitivity to SA and JA. I'm guessing that probably this will be a general stress response and that we see droughts and on high temperature that we might see an increase in RNA, but we've not tested that yet.
Now comes all the testing. Okay, we will stop here.
Lots
And
of great
Minu,
questions. Thanks, everybody.
there are many more, but Minu will direct those questions to you so people can
Okay.
get their answers connecting directly. Oh, okay, thank you very much. I don't know if Jean wants to say some goodbye words.
Oh yeah. Thanks everybody for coming to this event, and please watch for additional additional webinars in our series, and please alert the young investigators in your orbit, students and postdocs, to apply for the Rising Star series. That will be great to have more people applying. There'll be 3 chances to apply. So if you miss this round, you can apply later. Just keep watching our site and looking at social media for updates, and our website will be up very soon. Thank you, Roger. Yay. Awesome.
Thank you. Thanks, everybody.
We will see everybody again.
Bye.
Bye-bye.
Bye-bye.